Chapter name Cell Cycle and Cell Division. Introduction. Growth and reproduction are characteristics of cells, indeed of all living organisms. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cycles of growth and division allow a single cell to form a structure consisting of millions of cells. Cell division is of two types: mitosis, meiosis. Mitosis was proposed by Fleming. Mitosis produces genetically identical cells which are similar to mother cell. Cause of mitosis. First, kern plasm theory. Hartwig proposed kern plasm theory. According to this theory, mitosis occurs due to disturbance in karyoplasmic index Ki or nucleocytoplasmic ratio of cell. Karyoplasmic index Ki is equal to Vn by Vc minus Bn. Vn is equal to volume of nucleus. Vc is equal to volume of cells. Vc minus Bn is equal to volume of cytoplasm. Karyoplasmic index of a small cell is high as they have cell cytoplasm. Nucleus efficiently controls the activity of cytoplasm in small cells. In a large cell's nucleus fails to control the activity of cytoplasm. To attain the control of nucleus on metabolism, a large cell division into two cells. 2. Surface volume ratio. Surface volume ratio is also considered as a cause of cell division. When a cell grows in size, its volumes increase more than its surface, so a stage will reach when the surface area becomes sufficient to draw the material. At such critical stage, division of cell started. Cell cycle. Cell division is a very important process in all living organisms. During the cell division of a cell, DNA replication and cell growth also take place. All these processes, for example, cell division, DNA replication, and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesis the other constituents of the cell, and eventually divides into two daughter cells is termed cell cycle. All throughout cell growth, in terms of cytoplasmic increase in a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosomes DNA are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. Phase of cell cycle. A typical eukaryotic cell cycle is illustrated by human cell in culture. These cells divide once in approximately every 24 hours. Yeast can progress through the cell cycle in only about 90 minutes. The time period of cell cycle is varied from organism to organism and also from cell type to cell type. A cell cycle involves two stages: interphase, number 2 division phase या M phase. Interphase, this is phase between two successive M phase. In interphase, cell grows in size and prepare itself for next division. Interphase is most active phase of cell cycle. The interphase lasts more than 95% of the duration of cell cycle. A series of metabolic changes occurs during interphase in cell. These changes were not visible under microscope, so some scientists termed interphase as resting phase. It is the time during which cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. Howard and Pell classified interphase into three sub-stages: G1 phase और प्री DNA synthesis phase, first gap phase. G1 phase corresponds to the intervals between mitosis and initiation of DNA replication. During G1 phase, the cell is mitotically active and continuously grow. During G1, most of cell organelles increase in cell, and cell rapidly synthesize different types of RNA and proteins. Due to availability of protein, synthesis of new protoplasm take place in cell and it start growing inside. Cell grows maximum in G1 stage. Number 2, S phase, DNA synthesis phase. Replication of nuclear DNA and synthesis of histone protein and take place in S phase. Replication of cytoplasmic DNA may occur in any stage of cell cycle. During this time, the amount of DNA per cell doubles. If the initial amount of DNA is denoted as 2C, then it increased to 4C. However, there is no increase in the chromosome number. If the cell had diploid or 2n number of chromosomes at G1, even after S phase, the number of chromosomes remain the same, for example 2n. S phase marks the phase of DNA replication and chromosome duplication. DNA content in a chromosome become double. In animal cells, during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. Number 3, G2 phase. Second gap phase or post DNA synthesis phase, pre-mitosis phase. Final preparation of M phase occur during this phase. Special materials required for M phase are synthesized in G2 phase. For example, tubulin protein required for formation of spindle fiber. Cell growth continues. G0 phase. Some cells in the adult animals do not appear to exhibit division. For example, heart cells and many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. These cells that do not divide further exit G1 phase to enter an inactive stage, cell quiescent stage, G0 of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate, divide unless called on to do so, depending on the requirement of the organism. Checkpoints of cell cycle. Cell cycle is running by a group of special protein, cyclin and CDKS, MPF, Nasty Hunt and Hartwell 2001 studied on Saccharomyces. Number 2, division phase. Division phase or M phase or mitotic phase lasts for only about an hour in the 24-hour duration of cell cycle of a human cell. The M phase represents the phase when a actual cell division or mitosis occurs. In animals, mitotic cell division is restricted or only seen in diploid somatic cell except in some social insect. Against this, the plant can show mitotic division in both haploid and diploid cells. This is the most dramatic period of the cell cycle, involving a major reorganization of virtually all components of the cell. Since the number of chromosomes in the parent and progeny cells is the same, it is also called as equational division. Thought for conventions, mitosis has been divided into 4 stages of nuclear division. It is very essential to understand that cell division is a progressive process and very clear-cut lines cannot be drawn between various stages. The M phase starts with nuclear division corresponding to the separation of daughter chromosome, karyokinesis, and usually end with division of cytoplasm, cytokinesis. Mitosis is divided into the following four stages: prophase, metaphase, anaphase, telophase. Prophase, which is the first stage of karyokinesis of mitosis, follows the S and G2 phase of interphase. In the S and G2 phase, the new DNA molecules formed are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. The centriole, which had undergone duplication during S phase of interphase, now begins to move towards opposite poles of the cell. Anastral and amphistral mitosis. In higher plants, centrioles are absent and no esters are formed. Mitosis without esters is known as anastral mitosis. In animals, the esters are present and the mitosis is described as amphistral or estral mitosis. The completion of prophase can thus be marked by the following characteristic events: chromosomal material condensed to form compact mitosis chromosome. Chromosomes are seen to be composed of two chromatids attached together at the centromere. Centromere, which had undergone duplication during interphase, begin to move towards opposite poles of the cell. Each centrosome radiates out microtubules called esters. The two esters together with spindle fiber form mitotic apparatus. Cell at the end of prophase, when viewed under the microscope, do not show Golgi complex, endoplasmic reticulum, nucleolus, and nuclear envelope. Metaphase. In the complete disintegration of the nuclear envelope mark the start of the second phase of mitosis. Hence, the chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of chromosomes is completed and they can be observed clear under the microscope. This then is the stage at which morphology of chromosomes is most easily studied. At this stage, metaphase, chromosome is made up of two sister chromatids which are held together by the centromere. A small disc-shaped structure at the surface of the centromere are called kinetochores. These structures serve as the site of attachment of spindle fiber formed by the microtubules to the chromosomes that are moved into position at the center of the cell. Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator, with one chromatid of each chromosome connected by its kinetochore to a spindle fiber from one pole and its sister chromatid connected by its kinetochore to a spindle fiber from the opposite pole. The plane of alignment of the chromosome at metaphase is referred to as the metaphase plate. Chromosomal fiber discontinuous या kinetochore which run from pole to centromere and supporting fibers continuous या non-kinetochore which run from pole to pole arranged in cell. Centromere lies at equator and arm of chromosomes remain directed towards poles. The key feature of metaphase are spindle fibers attached to kinetochore of chromosomes. Chromosomes are moved to a spindle equator and get aligned along metaphase plate through a spindle fiber to both poles. Anaphase. Centromere of each chromosome split simultaneously. Lengthwise division of centromere. Sister chromatids separate from each other and separate each chromatid is now referred to as individual chromosome. Number of chromosomes become double in cell as each chromosome moves away from the equatorial plate. The centromere of each chromosome is towards the pole and hence at the leading edge, with the arm of the chromosome trailing behind. The two new daughter chromosomes begin moving towards opposite ends of the cell, and as their spindle fibers shorten due to depolymerization of tubulin protein towards kinetochoric end. Anaphase stage is characterized by the following key events: centromere split and chromatid separate. Chromatid, now referred as chromosome, move to opposite pole. Anaphase, telophase, interphase. Telophase, reverse prophase. At the beginning of the final stage of karyokinesis, for example, telophase, the chromosomes that have reached their respective poles decondense and lose their individuality. The individual chromosome can no longer be seen, and chromatin material tends to collect at each of the two poles. This is the stage which shown the following key events: chromosomes clusters at opposite spindle poles and their identity is lost as discrete elements. Nuclear envelope develop around the chromosome clusters at each pole, forming two daughter nuclei. Nucleus, Golgi complex, and endoplasmic reticulum reform.